Here are the safety measures that must be taken for this experiment. First, you must wear gloves and goggles. Second, the flash gel cassette contains a mutagenic agent that will bind to your DNA, so do not touch the actual gel surface of the flash gel cassette. Only the plastic casing of the gel should be touched when moving it. Third, the flash gel will run at potentially lethal voltages. Do not touch any portion of the flash gel unit while the power supply is on. Lastly, the UV light shield must be used to protect your vision. For this experiment, you will use a double-tiered flash gel cassette. Open the flash gel, pull the stickers off of it, and throw the stickers into the normal trash can. Do not place the stickers or the flash gel wrapper in the waste bucket on the side counter. Next, hydrate the wells by adding drops of RO water to each well. Carefully tilt the cartridge to drain the wells and blot the water from the edge of the cartridge with a paper towel. Slide the gel cartridge into place on the flash gel dock. The wells of the gel should be positioned at the left side of the dock and the center of the dock. Next, install the UV light shield by sliding it beneath the middle column of wells as shown. This is essential to your safety. If you turn on the flash gel light without the light shield installed, the UV light will damage your eyes. Also, keep the UV light shield after you're done using it. Do not throw it into the trash can. TAs. Before any of the student's DNA sample is loaded into the flash gel cassette, you will first load the DNA ladder. The DNA ladder is located in a foil pouch that looks like this inside of the refrigerator. Load 5 microliters of the DNA ladder into the flash gel. Make sure you do not have an airspace near the end of the pipette tip. Carefully insert the tip and slowly release the sample. Here we have loaded the ladder into the center wells of both columns. The student's DNA will be placed above and below the ladder. After you have finished loading the ladder, return the tube back to the foil pouch and return the pouch back to the refrigerator. Next, the students will load some of their PCR sample. Use a fresh micropipette tip and load some of your PCR sample into one of the lanes of the gel. Record in your lab manual what lane you load your sample into. Shown here is our loaded flash gel cassette. We loaded the DNA ladder into both well columns near the center of each column. The DNA ladder has a blue color. Also, we loaded our own PCR amplified DNA, which is labeled as student's DNA in this picture. The student's DNA is green in color. After all of the PCR samples have been loaded, your lab instructor will run the gel. First, plug in the electrodes as shown so that the color of the cables match the color of the sockets. Before you plug in the flash gel unit, make sure the UV light shield is in place as shown so no damage is caused to your eyes. Next, plug in the flash gel unit. After you plug in the flash gel unit, turn it on by pressing the orange light button. Next, plug in the power supply. Slide the power switch to the on position, slide the voltage switch to high, and turn the voltage control knob until the voltage reads 250 volts. Do not touch any portion of the flash gel unit while the power supply is on. Turn off the lights so that the gel can be viewed. Here we have fast-forwarded the video footage by 20 times. This gel actually took 3 minutes to run. Make sure that you turn off the power supply before the DNA runs off the end of the gel. When the DNA approaches the end of the gel, unplug the power supply. Turn off the power switch, slide the voltage switch to low, and turn the voltage control knob to minimum. Turn on the lights and unplug the electrodes. Also, it is now safe to remove the UV light shield, but do not throw it into the trash can because it is reusable. Set the box for the camera over the flash gel unit as shown, orienting the box so that the wires fit through the slot. Next, attach the camera with the camera hood. Turn on the camera and the settings should be as shown. Zoom in and take a picture. Because of the low lighting conditions, it is important that you don't shake the camera or you will get a blurry picture. When finished, dispose of the flash gel into the waste bucket on the side counter. Here's a review of the safety precautions you must take for this experiment.